And so let's get right to it. So our first speaker is Wendy Hwang-Verslues. Okay, good.
Perfect.
And she is coming from Taiwan. Wendy.
Thank you very much for the introduction. I would like to thank the organizer for this opportunity. Today I'm going to tell you about ovarian cancer. OC is one of the deadliest gynecological cancer in women worldwide. Approximately 50% of women would die within 5 years overall. But due to the aggressive nature of the disease and the lack of symptom, most of the patients were diagnosed at a later stage when the cancer cell has spread to distant organ. OC is hard to study and difficult to treat, majorly due to its complexity. It's really heterogeneous. Based on the origin, the molecular profile, and the histomorphology, they can be categorized into very different types of subgroups. High-grade serous carcinoma is one of the dominant one, which consists of 70% of the OC cases in the States and 30% of the OC cases in Asia. The most frequently mutated gene is P53. So most of the OC studies right now are focusing on this subtype. Therefore, the therapeutic strategy were also designed based on high-grade serous carcinoma. However, they cannot be as effective as for other subtypes. So today I'm going to bring your attention to the second dominant subtype, ovarian clear cell carcinoma, OCCC. OCCC has very distinct histomorphology, and most of the OCCC cases expressing wild-type P53, and the most frequently mutated genes are AR1A and PIK3CA. In the US and Europe, the incidence is usually below 10%, but in Asian countries it's higher and it's continuing increasing without known reason. In the early stage, OCCC is not particularly malignant. However, at advanced stages, they become more aggressive, more drug-resistant, easy to metastasize to different organs, as well as develop recurrence. And the 5-year survival at this stage is significantly lower than that of the high-grade serous carcinoma. According to the clinical observation, OCCC has higher frequency to form liver metastasis, and patients who develop the liver mets usually have significantly lower survival. So why OCCC is so malignant at advanced stage and why they can form liver mets so easily compared to other subtypes of ovarian cancer is not known. So we previously identified MEX3A as a critical oncogenic factor that drives wild-type P53 expressing ovarian cancer to undergo tumorigenesis. MEX3A is a dual functional protein that can bind to RNA to modulate RNA level, as well as modulate protein stability using its ring domain, which has the E3 ligase activity. So in OCCC cells, MEX3A can interact with wild-type P53 and facilitate its protein degradation. Therefore, depletion of MEX3A can increase wild-type P53 and induce ferroptosis in the cells. Therefore, in OCCC cells, when MEX3A is high, that can inhibit P53-mediated ferroptosis. More recently, we found that MEX3A has more function in addition to promoting tumorigenesis in the primary site. We found that MEX3A also contributes to liver mets. Depletion of MEX3A significantly reduces the liver nodules. And we further evaluate whether these differences in liver mass is due to the size of the primary tumor or the cell ability to propagate and colonize in the liver. So we first directly treated the cells with liver extract and found that MEX3A expressing cells are more resistant to the stress induced by liver extract compared to the MEX3A depleted cells. We also evaluated the anchorage independent growth ability, and we know that depletion of MEX3A can decrease this sulfur-alkyl forming ability of the cells, but liver extract further suppress such activity to a lower level. Interesting enough, that MEX3A expressing cells seem to enjoy such environment and can form more colony in sulfur-alkyl that contain liver extract. We then performed intrasplenic injection to bypass the early steps of metastasis and found that MEX3A expressing cells can colonize and propagate in the liver, whereas MEX3A depleted cells cannot. And this data really correlate with our clinical sample that when we compare the MEX3A expression in OCCC cells in various distant metastasis site as well as primary site, we found that MEX3A expression is highest in OCCC than in the liver. So together, this data really suggests that MEX3A is essential for tumorigenesis and liver mets for OCCC. So when cancer undergoes metastasis, it has to really constantly change their metabolism in order to adapt to different microenvironment. And since MEX3A is critical for primary tumor growth and liver mets, we hypothesized that it may play a role in metabolic reprogramming. We found that MEX3A depleted cells has mitochondrial fission phenotype indicated by the elevated level of MFF and phosphodrip and their colocalization in the mitochondria, which will make the already elongated mitochondria from the MEX3A expressing cell become fragmented in MEX3A depleted cells. And since MEX3A depletion can increase wild-type p53, and p53 is known to participate in mitochondrial regulation, we generate the double knockdown cells and found that even depletion of p53 in MEX3A knockdown cell cannot rescue the fission phenotype or the fragmented phenotype, suggesting that this MEX3A mitochondrial regulation is p53 independent. So mitochondrial fission is important when cells undergo division, but chronic fermentation can be detrimental because it will lead to impaired mitochondrial function, which is what we found. In MEX3A depleted cells, with or without p53 expression, the mitochondrial membrane potential was significantly decreased, superoxide level was significantly increased, and NAD and NADH level was decreased, which suggests that the respiratory chain may have some problem. Indeed, functionally, in this MEX3A depleted cells, oxphos activity was significantly inhibited, which led to a reduction of ATP level. We further performed gene set enrichment analysis using our RNA-seq data, and we found that 30% of the genes that in the oxphos cancer hallmark were perturbed in MEX3A depleted cells. Differential gene expression analysis further showed that in MEX3A depleted cells, these oxphos genes are globally downregulated, including several genes that encode complex 1 subunits. Therefore, in MEX3A depleted cells, there's a defect in supercomplex 1 assembly. So during metastasis, the cell has undergone metabolic reprogramming, and mitophagy is one of the key processes that can get rid of the mitochondria the cell do not need or remove the damaged mitochondria that caused by the stressors in the microenvironment. And once the damaged mitochondria is removed, the cell has to be able to generate new and healthy mitochondria, or the cells will eventually die from depleting their remaining mitochondria. So we found that in MEX3A expressing cells, with or without liver asteroid, that will not affect the proliferation or survival of the cells. However, liver asteroid can severely make the cells undergo cell death within 3 days time course in liver asteroid treated cells condition. So since MEX3A is important in regulating mitochondria in OCCC, we think that maybe something in the liver asteroid can cause mitophagy stress to the cells, and MEX3A depleted cells just cannot recover from it. So to test that, we first treated the cells with liver asteroid, and we found a induction of mitophagy as well as autophagy in the control cell. But in the MEX3A depleted cells, probably due to the fermented mitochondria, the stress marker is already high in the basal level. But a liver asteroid can further induce the autophagy signal here. Importantly, after 12 hours in the liver asteroid containing media, we see a dramatic decrease of time 20, indicating that the cell is actually depleting their mitochondria. But there are a lot of stuff in the liver asteroid that can cause several different kinds of stress to the cell. We then directly treated the cells with FCCP to depolarize mitochondria to induce mitophagy stress, which can be indicated by these stress markers. What I want to show you is that in the MEX3A depleted cells, after FCCP treatment, the autophagy was induced. But then 12 hours after the FCCP treatment, all the mitochondrial protein were basically gone. It's hard to detect, suggesting that the damaged mitochondria was removed, but MEX3A depleted cells cannot produce healthy mitochondria to help the cells to survive, which reflect in this cell survival analysis. Within the 3 days time course, we did not remove FCCP, and you can see that at the end of the time course, 50% of the MEX3A expressing cells can still survive. However, the MEX3A depleted cells are basically completely dead. So this data basically tell us the MEX3A control of mitochondria is critical for the cells to recover from mitophagy stress. So with all this data, that led us to propose this model, that when OCCC undergo tumorigenesis and metastasis to liver, if the cell contain MEX3A and express a high level, they have functional mitochondria, they have functional oxphos, they can recover from mitophagy stress. So they can grow bigger, and they can probably survive better in the circulation and reach the liver and form nodules in the liver. However, if MEX3A is depleted, these cells will have fermented mitochondria, impaired oxphos, and they cannot recover from autophagy stress. Therefore, they form smaller tumor. If some of them can escape and go to the liver, they can barely form nodules. So I think that the study really identified MEX3A as a very important factor that drives OCCC tumorigenesis and liver mets. With that, I would like to acknowledge the student who did all the work that I presented today and the funding sources from Academia Sinica and the National Science Technology Council in Taiwan. While you're looking at this beautiful skyline in Taipei City, I would like to thank you for your attention and take any questions.
Okay, first question goes to our organizer.
Hey, beautiful talk.
Thank you.
So I have a question regarding the specificity for OCCC, because the question I have is this a mechanism that you would expect to occur in various types of cancer types that have wild type P53?
That's a good question. The answer is I don't know, but I hope so, because it can generalize this phenomenon. Why I say that, I will jump to the next slide to show you, because on the right-hand side there, there's a high-grade serous carcinoma. So people have found that MEX3A also promotes tumorigenesis in high grade, but their genetic background is very different, and their behavior is quite different. And it seems like MEX3A is actually having this context-specific function. So instead of regulating the mitochondria in high-grade serous, MEX3A depletion didn't cause any problem in this mitochondrial regulation. So we think that if, let's say, maybe kidney cancer, because they do have the clear cell subtype and they have similar pattern, maybe something like that will show the similar phenomenon, but we don't know yet. Thank you.
Can you comment maybe quickly on what you think it's actually doing in a complex, specific way to prevent or maintain mitochondria, or it seems like actually prevent excess mitophagy?
Yeah, this is very interesting because I think there are several layers of the questions. So MEX3A is RNA binding protein, but it also has the E3 ligase. So at the time that we saw this mitochondria regulation, we're wondering whether it can go into mitochondria and do something. But then it doesn't have the mitochondrial signal peptide in the protein. But we found that it actually colocalized in the mitochondria. And when we did a lot of biochemical assay, found that actually it's inside mitochondria. So why? We don't know. And we're thinking like maybe in OCCC there's some specific protein or chaperones that actually bring the MEX3A in there. And once in there, it actually bind a lot of proteins that involve in the respiratory chain and complex 1 biogenesis. So it's actually interesting that we're trying to figure it out.
Yeah, it might be cool if mutations could separate the E3 ligase from
yeah,
the
exactly.
RNA binding.
Thank you.
I was wondering if you looked at the activity of mitochondrial biogenesis master transcription factors like PGC1 alpha or TFAM that might explain this inability to recover mitochondria after
Yeah, thank
mitophagy.
you. We're actually doing that right now.
Okay, great. Thank you so
very
much.
much.
Let's move on.